Let's rewind the clock to early 2021. The world was in a frantic race against time. Yeah, the first COVID-19 vaccines were just starting to roll out, which was amazing news. But for the millions of people who were already sick, the question was much more immediate. What about a treatment? How do you actually fight a virus once it's already in your system? We're going to dive into that global hunt for a cure, exactly as it stood at that critical moment, all based on a really comprehensive scientific review from that time.
To really get a sense of the urgency, you have to look at the scale of the problem. By the beginning of February 2021, this was the number of confirmed COVID cases around the world, over 102 million people. And, that isn't just a statistic. It was a massive global crisis that demanded an immediate, all-hands-on-deck scientific response.
All right, so here's how we're going to break down that global strategy. First, we'll get to know the enemy, the virus itself. Then we'll look at this clever idea of repurposing old drugs for a new fight. After that, we'll explore the two main battlefronts, attacking the virus head-on and also trying to calm down the body's own chaotic response. And finally, we'll see how supercomputers got in on the action and figure out exactly where things stood in early 2021. Okay, so before you can treat anything, you have to understand what you're up against. For scientists, job one was to map out the SARS-CoV-2 virus, figure out its structure, its life cycle, and most importantly, find its weaknesses. The virus's game plan is actually pretty simple, but it's incredibly effective. Just think of its spike protein as this perfectly cut master key, and that key is designed to fit one specific thing, the ACE2 receptor, which is basically the lock on the outside of our own cells. The second that key turns in the lock, the virus is in. From there, it releases its RNA, which is literally the instruction manual for how to build more viruses, and it hijacks our cell's machinery to start churning out millions of new copies of itself. So here's the problem. Developing a brand new drug from the ground up can take more than a decade. And in a pandemic, you just don't have that kind of time. This reality forced scientists to get creative, which led them to a really crucial strategy: drug repurposing. Drug repurposing is all about teaching old drugs new tricks. Scientists essentially scoured the world's entire medicine cabinet, looking at drugs that were already approved for things like malaria, Ebola, or HIV, and they were hunting for anything that might just happen to work against this new coronavirus. And the huge advantage here? These drugs had already gone through safety trials in people, which meant you could potentially shave years off the development timeline. But, and this is a big but, this strategy was no magic bullet. As the paper this is based on puts it, repurposing was a double-edged sword. On one hand, it offered the tantalizing promise of a quick fix. But on the other hand, a drug designed for something else is rarely going to be a perfect match for a new virus. And that can lead to, it not being very effective or even causing some really dangerous side effects. It was a huge, high-stakes gamble. All right, let's shift gears and look at the first major battlefront in this war, launching direct assaults on the virus itself. The goal here was simple: find something, anything, that could stop SARS-CoV-2 dead in its tracks. To do that, scientists zeroed in on three critical moments in the virus's life. Think of it like a military operation. You've got a few options. You could block the invasion right at the gates. That's stopping its entry. Or you could let them in but sabotage all their supply lines and communications. That's messing with replication. Or your third option is to stop them from sending out new troops, and that's blocking the assembly and release of new virus particles. Early on, a lot of hope was pinned on old anti-malarial drugs like chloroquine. The theory was that they could somehow mess with the ACE2 lock on our cells, making it harder for the virus's key to work. But by early 2021, the trials had come in, and, well, it turned out they didn't really work. And what's worse, they could cause serious heart problems. A much more promising approach was to target a human enzyme called TMPRSS2. You could think of this enzyme as a tool the virus absolutely needs to prep its key before it can even try to unlock our cells. So if you block that tool, you could stop the whole invasion before it even starts. Okay, so what if you can't stop the virus from getting inside? Well, maybe you can stop it from making copies of itself. And that brings us to the virus's personal photocopier, a little enzyme called RNA-dependent RNA polymerase, or RDRP for short. Its one and only job is to read the virus's RNA instructions and just churn out endless copies. And this is where remdesivir, a drug that actually failed in trials against Ebola, suddenly became the frontrunner. It works by pure deception. Remdesivir basically puts on a disguise and pretends to be one of the building blocks for RNA. So when the virus's photocopier, that RDRP enzyme, grabs it to make a new copy, it's like grabbing a piece of paper that's secretly coated in glue. It just gums up the whole machine and grinds replication to a halt. Back in early 2021, this was by far the most promising direct antiviral we had. Now, the final direct attack strategy was all about sabotage. See, after the virus makes all its proteins, they come out in these long, useless chains. A special viral enzyme called a protease has to come along and snip those chains into their final working parts. So drugs like lopinavir and ritonavir, which were originally made for HIV, were designed to block that protease, basically shutting down the virus's entire assembly line. But here's the thing, just stopping the virus was only half the problem. It turned out that in the most severe cases, the biggest danger wasn't the virus itself. It was the body's own chaotic, out-of-control response to the infection.
This question became absolutely central to treating severe COVID-19. What do you do when the very system that's supposed to protect you just goes haywire and starts doing most of the damage itself?
This is something called a cytokine storm. Normally, cytokines are these really helpful little signals that coordinate your immune system's attack. But in severe COVID, the body can just dump an uncontrolled flood of them into your system. This storm triggers massive, widespread inflammation. It fills the lungs with fluid, and it can lead to catastrophic organ failure. So how do you calm a storm like that? Well, scientists were testing a few different tactics. Some drugs, like tocilizumab, act like signal blockers. They specifically target and neutralize a key inflammatory molecule. Others, called JAK inhibitors, work from inside the cell to basically turn down the volume on the whole cytokine alarm system. And a third approach was to use steroids, powerful general-purpose anti-inflammatories, though their use was pretty debated because they can also suppress the helpful parts of the immune response too. Okay, with thousands of potential drugs out there and dozens of different parts of the virus to target, this whole search was like trying to find a needle in a global haystack. To speed things up, scientists turned to a very powerful ally, the supercomputer. This whole approach is called an in silico study. It's a fancy way of saying in silicon, as in a computer chip. The idea is, instead of spending months and months physically testing thousands of drugs in a wet lab, researchers built these incredibly detailed 3D models of the viral proteins. And then they used massive computing power to simulate how thousands of different drugs might physically lock onto and disable those proteins. The logic behind it is actually pretty simple, but it's incredibly powerful. A computer could identify a critical piece of the virus, like the 3CL-Pro protease that it needs for assembly. Then it would just zip through huge databases of known drugs, digitally checking to see which molecules had just the right shape to clog up that target. This just dramatically sped up the whole process, letting scientists pinpoint the absolute best candidates to then test in the real world. So after this absolutely frantic year of research and repurposing and clinical trials, where did the quest for a COVID-19 treatment actually stand at the beginning of 2021? What did we learn? This scorecard pretty much sums up the state of the race back then. The direct antiviral remdesivir was definitely showing real promise in helping patients get better faster. At the same time, all that initial hype around chloroquine had completely fizzled out. On the immune side of things, drugs like tocilizumab were proving to be really powerful tools for the sickest patients. And other supportive therapies, like giving patients plasma from recovered people or using anticoagulants, were also carving out a crucial role in helping people survive. You know, the most important lesson from that first year was crystal clear. There was not going to be a single silver bullet. The dream of finding one miracle drug that would solve everything was replaced by a new, more complex reality. The best path forward was a multi-pronged attack, combining drugs that slow down the virus with therapies that help the body survive its own fight. And that leaves us with the big question, doesn't it? The science of 2021 proved that hitting the virus from all these different angles was the best strategy we had. But in a long-term fight against an enemy that is constantly changing and mutating, can that kind of complex, multi-front attack ever truly lead to a checkmate? That was the challenge then, and really understanding that race gives us some critical insight into how we're going to face the pandemics of the future.